Hi everyone, today I want to share with you my roadmap to writing a scientific manuscript and never getting stuck in the process. I will break down which sections you should do first, which should follow, and why. To get into the details, I write a tiny paper with you to show you exactly how it works. So let's dive into it. Okay, the place where I always like to start my papers is with the panels and figures. So the first thing that I like to do is to sketch all of the panels. So for example, here we have 4 panels. The first one is an RNA-seq, where we have mRNA levels here for a control. And in this axis, we also have mRNA levels for the experimental. And then we have a bunch of transcripts and we have a lot of downregulated here. So a second experiment that we did was, for example, a gene ontology, GO, and we found that from this bunch of downregulated genes, the most enriched term is protein localization, and this is going up significantly. Then we can have RNA metabolism, this is going up, and then we can have something like spermiogenesis, also going up a little bit. Then in this manuscript, we have a third panel when we do a Western blot of protein alpha corresponding to this alpha transcript that is very downregulated. So we have the Western blot, we have GAPDH as a control, and then we have our protein alpha that is abundantly expressed in the controls, and in the experimental is completely or almost entirely gone. And finally, in this tiny paper, I have this panel here where we are doing an IF. So here we have one cell with the nucleus, the other cell in the nucleus, and we are going to do an IF for a protein T here that we stain in green. So we have the control, we have the experimental conditions. In the control conditions, we have the localization of the protein here in what we can call the Golgi, and then the experimental we have much more diffuse through the whole cytoplasm. These are the experiments that we did in this tiny paper. For now, it's difficult to understand what is the narrative, so this is what comes next. Okay, now that we have the panels, we need to create a narrative where we explain each panel and link them sequentially in a linear story. So the first section I want to address now is the results. This is the heart of the manuscript and everything will follow from here. So for the results, we will explain each of these panels individually and we will provide the minimum background we need for each of them. So let's start with this RNA-seq. Here we explain that protein X, that is our protein of interest, is required for the liver to function, and we give a reference here. And then we say protein X is an RNA binding protein that has been shown to increase the stability of mRNAs, and we give the reference. So this will be our experimental, will be the knockout of that protein X. So it's minus protein X. And we are doing an RNA-seq, and this is consistent with protein X being an RNA protein that promotes the stability of transcript. So the question that we want to answer with this, or the point that we want to address, follows. So we say to investigate the role of protein X in liver, we did RNA-seq of primary hepatocyte knockout for gene X and controls. And then we explain what we observe here. So we say we observe 100 significantly downregulated transcripts and no significantly upregulated transcripts. So this is just the description, and we say that this is in figure 1a, so that's the result. And finally, we give a little conclusion for this panel. We say this observation supports the model in which protein X acts as an RNA stability factor in the liver. So this is the mini conclusion. So we are done with panel A. Now we need to move to panel B in the same way. So let's go through these 3 panels that are remaining just to have an idea of how the whole process works. So now if we move to the gene ontology, we can say, okay, to understand what mRNAs are stabilized by protein X, we perform a gene ontology analysis of the significantly downregulated transcripts, and then we explain the results. Genes regulating protein localization were significantly enriched in our datasets, followed by transcript relevant to RNA metabolism and spermiogenesis. So this is the description of the results, and we say these are in figure 1b. And finally, we give a little conclusion here. But in the first sentence, we provide a little bit of background. So we say protein localization has been shown to be critical for pancreas homeostasis. This is just to bring a little bit of extra context that we didn't have when we started thinking about the manuscript. And then we conclude the results of this panel here. So we say, thus this data suggests that altered protein localization in the hepatocytes can also be a driver of liver failure. And that will be the conclusion for this panel. So now let's move on to this Western blot. So here we start by saying, in order to understand how protein mislocalization affects liver function, we examine the list of most downregulated genes, and gene alpha here is the most downregulated transcript in the transcriptome in the absence of gene X, and this is the back to the figure 1a actually, not B. And then to determine whether the downregulation of the mRNA leads to a reduction in protein levels, we perform this Western blot that we are showing here, and then we explain the results. Consistently with the decrease in transcript level, we observe an almost complete depletion of the alpha protein. And the conclusion is that thus protein X appears to promote the accumulation of protein alpha in the liver by stabilizing its mRNA. Okay, and with that we move to the last panel here, the IF. So we explain this panel in detail. So here we start by introducing some background that we have. So we know that gene alpha has been shown to anchor protein T in the Golgi in the pancreas, and this is previous work that we are referencing. And then we say mislocalization of protein T in the pancreas leads to pancreatic dysfunction. This is another set of manuscript, for example. So here what we want to understand, this is the question that we want to answer. Then we ask to determine whether protein T is mislocalized in the liver without protein X. We examine its localization by IF in protein X depleted hepatocytes and controls. So now we describe what we see, and we have while protein T is localized to the Golgi in white blood cells, protein T is present throughout the cytoplasm in protein X depleted cells. So this is the result, and now we make a mini conclusion for this panel. So this data supports a model in which protein alpha anchors protein T to the Golgi in hepatocytes. And this is a general conclusion. We say the mislocalization of protein T can explain the liver damage in X-depleted hepatocytes. Okay, now that we have the main narrative, we want to move to the introduction, where we will put together all the background used in the results. So let me show you how this works. So if we go to the results sections, we can sort of highlight different regions here in pink that are introductory, and in green here are regions that are more conclusion. So for the first paragraph, we are introducing that protein X is required for the liver, and that protein X is an RNA binding protein that promotes stability. So that's the main question that we want to answer, or the main topic that we want to study. So we start by introducing the topic, the problem, which is related to these first references. So we can say mutations in gene X are associated with higher incidence of liver failure, and we give here the same reference. Here is like we are stating the problem. The next thing is just to define the question or the gap in the knowledge. So this is, however, the role of protein X in liver function is not known. And then we continue giving a little bit more the information about what protein X is doing. So we say X is an RNA binding protein known to stabilize transcripts by binding 2-3 OTR, but the only study so far has been done using immortalized cells. And we give here the reference to that work. So that concludes the first paragraph of the introduction. We use whatever we were using before to introduce that first panel. And now what we can do in the result is probably remove some of those sentences if they are very repetitive, and this will facilitate the reading of the results. We will go faster to the data without dwelling too much into the introduction that will be already explained in the introduction itself. So while we were describing the panels, we stumbled into this issue of protein localization. Here, don't worry too much if there is some sort of jump between the narrative. Here we are introducing the different characters in this story, and it's fine if in the introduction we do some cutoff between the different topics. So we can introduce protein localization, but saying protein localization is critical for the proper function of the cells, and we give some sort of review, and we go more specific. In the pancreas, the protein alpha has been shown to anchor antigen T to the Golgi membrane, and we get there the references. Note that this is all the information that will be required to know to explain each of the panels, so we are making sure that whatever we use to explain the panels appears here in the introduction. And then we can go into mislocalization of T antigen in the absence of protein alpha leads to the depletion of insulin producing cells in the pancreas, and we give here the references. So here we completely introduce the topic of protein localization and this relationship between the protein alpha and the antigen T localization. The final paragraph of the introduction is more or less a summary where we introduce what we are addressing in the paper, and we give the methodology and the basic results we obtain. So here we can write, here we study the molecular function of protein X in hepatocytes, and then we say using RNA-seq we found that the protein alpha is significantly downregulated in the absence of protein X, and then when we examine the distribution of protein T in hepatocytes by AF, where the levels of protein alpha were significantly reduced, we observed that protein T localizes throughout the cytoplasm in the mutant cells, in contrast to the discrete localization to the Golgi in wild-type cells. And then we conclude this observation suggests that protein X stabilizes the mRNA of protein alpha, which in turn anchors protein T to the Golgi of hepatocyte cells and promotes hepatocyte function. Finally, we can move to the discussion where we put together all the conclusions from all the results and we propose the most likely explanations. So again, in the results we have all these final sentences where we have been sort of concluding the main message of each result, but these sentences here only apply to that panel. In the discussion we try to put them together, and in the first paragraph typically we go and we give a summary of the main findings. So in this case we say, in this study we describe protein X as a primary factor contributing to protein localization in hepatocytes, and we say by sustaining the levels of protein alpha. And then we try to bring together the different results from the different sections. So we say the decrease in alpha mRNA and protein levels indicate that protein X acts on the mRNA, although we cannot rule out the contribution of protein X on the translation of transcript alpha. The sharp decrease in transcript levels suggests that the stabilization of the transcript is the primary regulatory mechanism. So here in this case we try to explain why we think our model is the most simple and therefore more likely with the evidence that we have at hand. So this is something important that we can do in the discussion. So in another paragraph of the discussion we can go into more detail about the issue of protein localization, and here we can also put our study into a broader context. This is another important thing that we can do in the discussion. So in this case we can say protein localization has been increasingly recognized as a critical aspect of cellular function, And then aberrant protein localization has been shown to contribute to pancreatic dysfunction. And we give again these references. And here we say, okay, our data suggests that similar factors regulating protein localization in the pancreas are altered in our liver disease model. And the extent of pathologies related to protein localization remains to be determined. So here we are putting our findings into a bigger context, that this is also important to do in the discussion and not in other parts of the manuscript. So a final thing that we can do also in the discussion is to leave open questions that we haven't addressed. In this case, we can write mislocalization of protein T leads to pancreatic cell death. Similarly, the mislocalization of protein T likely explains hepatic dysfunction in cells depleted with mutating protein X. But then we bring the question, however, we cannot exclude the contribution of other altered pathways because there were several genes regulated by protein X. So we say we envision that protein T localization in the liver can be used as a marker of protein dysregulation in the clinic. So here we can also say, okay, how can we expand the results of our study beyond our specific field? So once you have the figure, results, intro, and discussion, you can move to the rest. If you want to dive more in depth into each of these sections, please check my playlist on how to write a manuscript. Okay, that's it for today. We just covered how to use your panels and figures to guide your results narrative, and then how to build your introduction and discussion based on your results. If you like this content, please subscribe to the channel. It is the best way to make these videos more accessible to other people like you, including yourself in the future, and I will see you in the next video.